toxins. So they're toxins that are produced by these bacteria, and they act on the gastrointestinal system. In the case of cholera, this has been pretty well documented. Cholera typically kills people by dehydrating them. And so what happens is the cholera toxin causes your gut, instead of your large intestine, normally the primary function of the large intestine is to absorb water. Pull that water out because you dump a lot of water into the lumen of the GI tract in order to digest properly. You make a kind of soupy mess of food material that's being digested. When it gets to the large intestine, the primary function is to take that water back out, absorb it back in the bloodstream. Under the influence of these toxins, instead of absorbing water, the gut secretes electrolytes into the lumen. The water follows, and the person loses water. They suffer from severe diarrhea, and they lose so much water that they can become dehydrated and die. Now, we think of that primarily as being a characteristic of cholera, but Escherichia coli can do the same thing. And Escherichia coli is a very significant pathogen in many parts of the world because it's in water. And unfortunately, parents feed their children, their infants, water that has Escherichia coli that produces these toxins that can then cause severe diarrhea. And the infants can become dehydrated to the point of being, the dehydration is fatal in a very short period of time because they're so little. So Escherichia coli kills infants. It's especially problematic because normally mothers would breastfeed their infants, but in some countries breastfeeding is considered, well, maybe you shouldn't do that. Instead, you should feed infant formula. The idea is maybe that's better for your baby. It's definitely not if the water supply is not clean. And so they feed them Escherichia coli and they die. And of course, Shigella causes severe dysentery and also diarrhea. Very similar types of toxins that these organisms are producing. So there are some toxins produced by certain organisms, such as Corynebacterium diphtheriae and Pseudomonas aeruginosa. Again, these are toxins that are released into the environment, but they don't act on a specific type of tissue. In general, these prevent protein synthesis. The Corynebacterium diphtheria organisms colonize the throat. So if a person gets diphtheria, they don't have the Corynebacterium throughout their system. They get the Corynebacterium in their pharynx. They live in the pharynx, they release these toxins into the bloodstream, and it's actually the toxin that causes diphtheria, which is why when you get the Tdap, you're getting the diphtheria toxoid, which is going to neutralize, you're going to produce antibodies that will neutralize the toxin. Pseudomonas aeruginosa, most of the pictures that you see there are Pseudomonas. There was an extra credit question up here asking, what is that blue-green pigment? It's called piocyanin, and it is a characteristic pigment produced by Pseudomonas aeruginosa that allows that to be recognized when it's in association with, in that case, an eye infection or an ear infection, or sometimes you'll see it around a catheter where a catheter is in place, or you can see it in burn wounds where there's this green coloration. At any rate, Pseudomonas aeruginosa is also problematic because they produce these toxins that prevent protein synthesis, and they're especially problematic in the case of burn victims because they can't grow new skin, they can't heal because they can't synthesize, their cells can't synthesize proteins. Then there are toxins that are called endotoxins. So it said entero were exotoxins, but they acted on the gut. The endotoxins, this is our alternate category. These are not proteins that are released by living cells. These are substances within cells, and they're typically only released when the cells die. So killing the cells will release these. When we were talking about gram-negative cell walls, and I said the gram-negative cell wall has an outer membrane. That outer membrane has complex composition. One of the components is called lipopolysaccharide, typically abbreviated LPS. The lipopolysaccharide portion of gram-negative cell walls is an endotoxin. It's released when the cells die. And unfortunately, the endotoxins tend to have a wide range of effects within the body. So they're not like the exotoxins that have a specific target and a specific action. Instead, they interact with a whole bunch of cells and they bring about a lot of changes. So they react with these receptors that are called toll-like receptors. We didn't talk about toll-like receptors as part of the immune system, but lots of cells, including macrophages and B lymphocytes and mast cells and epithelial cells, have these toll-like receptors on their surfaces, and so they can bind to those endotoxins, and then those cells become activated. If they're T cells, they're going to release cytokines. If they're macrophages, they're going to become active and they can release complement factors, and they can release lysozyme, and they can begin to cause some damage. They will often cause cell death. They can be triggering the release of interferon, but oftentimes you get response kind of like body-wide inflammation. So it's more like anaphylaxis, where a person goes into circulatory shock. And sometimes if you have E. coli in the bloodstream, treating the patient and killing the E. coli will actually trigger a worse response because of the endotoxins that are part of the E. coli wall. So you get elevated temperature, you get circulatory malfunction, general feeling of malaise, and as I said, circulatory shock, which can be deadly. This goes into some important etiological agents. I don't want to go very far with this, but I wanted you to know that as we start talking about the etiological agents, you're going to have on the final a large matching section. So you've been studying as we go through the semester, you've had these outbreaks. Most of the outbreak organisms are going to be on this matching, where you have to match the genus. Genus names are in one column. With species names are in a different column. With the disease names are a third column. I had to split them out that way because many of the species names, like tuberculosis and pneumonia, tell you what the disease is, so I put the genus separate from the species. So be ready for that on the matching. But there are 3 organism names that I want you to know that are not going to be on the matching, and these are Staphylococcus aureus, Escherichia coli, and Pseudomonas aeruginosa. When we were doing our antimicrobial sensitivity testing, and we had those 3 up on the board, as I said, at various microbiology conferences, the microbiology conferences are often combined with people that are associated with just the American Society for Microbiology, but also the society involving clinical microbiologists. And those 3 organisms are recognized as being the most common encountered in hospitals locally. And they are potentially deadly. Staphylococcus aureus can kill people. Escherichia coli can kill people. Pseudomonas aeruginosa, unfortunately, kills mostly children with cystic fibrosis because they get into the lungs and you can't get rid of them. They just keep, you can treat them and kind of knock their numbers down, but they just keep coming back. So you need to know those, and I'll go over some specific characteristics of them, but you're not going to see those on a match. You're going to have to write them out. Practice spelling them. Yeah. Don't confuse Staphylococcus and Streptococcus, on my word. Staphylo is great blood clusters. Strepto is chains. Not the same thing. So yeah, Staphylococcus aureus, Escherichia coli. Will I accept E. coli? It's like, oh,
yeah.
Yeah.
How about Chipotle disease?
No, you can't call it Chipotle disease or Jack-in-the-Box disease or those other things. No. Okay, so we'll do the etiological agents next time. So now you can run away and rest.
Rest until
You act like we don't have more to do.
I know you have more to do, but you don't need to do it right now.
We do, we don't have time.
Huh?
I found the second page.
Oh, all right. In that case, I'll have to accept it.
What? Remember the glycolysis homework? I forgot the second
You
page.
forgot the second page.
You forgot the second
It
page?
was only
Yeah.
3 points.
That's only.
That's dedication.
Wait, so
Still
it's,
look.
So you would have got a 20 out of 20 if you actually
Yeah.
turned in the back page.
or 92.5.
Because she's awesome. She's going to give us 3 extra points.
What a nice person, I wouldn't
I'm
know.
at a 92.5.
I
That's what I'm
know.
asking.
Yeah, 92.5 is an A.
It's weird.
How about 92.1?
92.1.
Because on
Well,
the
if you get like a
syllabus,
92.4.
on the syllabus it
I
says
really don't want to be
You're
there.
one of
on the
those.
syllabus it says
see this
80 to
part.
92%.
Yeah.
92 to 93% is undefined.
Why? You don't need it?
She that's wiggle room. This, how good and how much she likes it.
How much was that That's helpful and
wanted to ask
And how much was it due to anatomy?
Well, I can't take that into account. I can tell you that I only once gave a B to a student with a 92. And I didn't feel good about that.
Were you doing it during that?
Yeah.
You gave him a B?
I did after the test.
Only one time did I do that. And afterwards, and it's because, look at the rest, if I was grading on a curve, that wouldn't even be close to me. Look at our high score. I know you don't wanna look at them,
I
but
don't.
have
I'm
to
only
look
worried
at
about mine.
them. I have to say, what does an A mean? It means outstanding, and our outstandings are really high. So you don't wanna be on that border.
But why are we comparing each other? We're all individuals.
There's only one time to ever give an
Well, I don't want to, that's why I try to go by straight percentage.
Okay.
I know. I know. I know this. I'm sweating balls.
Alright, I'll see you later.
Good. Does your girlfriend hate us?
Huh?
Does your girlfriend hate us?
No, she actually likes you
Does
guys.
she think I'm gonna stab her because she's a nursing
I
leader?
like your friends.
Yeah.
There you go.
Literally.
Clear,
good.
clean picture.
Yeah, that's good. Yeah, no, she really did say that.
I took this today
She's nicer
when
than
I was
Jaylena.
coaching. I took that when I was
Jaylene's
coaching.
like, if some girls come up to my boyfriend, I would just not even ask them. Just
I don't know if the
Yeah.
like the sevens can do this, but this is kind of dope
No, just
because
after.
you can do like different things, so like, you can loop it.
That's
One
pretty
minute read them all, I guess.
Where's that, Sierra? Or is that
This was at Lifetime,
Nice.
where you can do like you can just long exposure it, so just blend.
There's one that just says.
that's-
I thought this was like a kind of like a cool touch though,
Yeah.
because I've never had the live photos either.
Yeah, oh yeah.
So this is like all
That's
new,
true,
like
that's
cool
true.
stuff, man.
That's one of the other ones over there. I did.
No,
Is the
What
one
is
that
the
has
and on
later.
top say?
You do.
There's lots of couple words on top.
How could you not?
I don't know.
Oh, man.
Cells slash tissues.
No, I can't. I can't let you do that.
Slash.
do
I
what?
know there's all kinds of
Translate
communication that goes
cells
on.
fat slash tissues.
I didn't agree. Fair, touche.
What'd you do, DJ?
She
Didn't read something.
I think I have it.
there was. You were right.
I can't read it. He's the tumor root.
The calculator?
Like NEC. And 2 ER, not a K, right?
Let's do it. You can cheat on your girl.
Let's do it, bro.
I'm scared to try and write it down, but you know what?
Oh, we gotta restring our plates real quick because we're probably out of time this week.
We'll just restrict it on nutrients. I don't want the fungus. I don't want you to play with that. You come to me and open that. I know we wanted to try it because I wanted to see if they produce a dark red. They don't on here. Because the one that has a description, it says they're dark on the back side, and yours aren't. They're not on here either. You can see them growing on them. Nobody else. So they're not, which makes me think maybe they're the second one.
Yeah.
There were 2 on the top of the list, and one of them came from some lakes. This is the lake one that said it's distinctly different from the other one.